So hello everyone and welcome to today's webinar hosted by Kapa Biosystems. My name is Andrea Carbone and I'm the marketing manager and moderator for this event. So before we begin, I'd briefly like to go over some GoToWebinar features to help ensure everyone enjoys our presentation. So all attendees have been muted for the call to provide the best sound quality. However, we will be taking questions, so please feel free to type them in the questions panel throughout the presentation, and we'll attempt to answer requests at the end of the webinar and within our 1 hour allotted time frame. Today's presentation will be recorded and hosted on Kapa's website later this week, and an email notice will be sent to all attendees. So Kapa is proud to host Dr. Jonathan Keats from the Translational Genomics Research Institute for a special webinar on evaluating library prep workflows for patient-related genomic research. Dr. Keats is highly respected in his field of multiple myeloma research and understands the importance of rigorous evaluation of library prep solutions to capture the highest quality sequencing data, since the outcome directly affects patients' lives. Dr. Jonathan Keats' lab at TGen is focused on using state-of-the-art methods to comprehensively investigate the genomic features of disease, with the goal of identifying genetic events that drive the development, progression, and mediation of therapeutic resistance. His group currently oversees the genomic characterization of patients in the MMRF COMMpass trial, where comprehensive analysis is performed on each tumor and matched normal sample using long insert, whole exome, and RNA sequencing. Dr. Keats is primarily interested in multiple myeloma with secondary interests in other hematological malignancies and immunodeficiency syndromes. He received his PhD from the University of Alberta in 2005 and did his postdoc at the Mayo Clinic. In July of 2010, Dr. Keats joined the Translational Genomics Research Institute, a nonprofit organization in Phoenix, Arizona, where research is focused on helping patients with diseases such as cancer, neurological disorders, and diabetes. Recently, he was intimately involved in the Multiple Myeloma Genomics Initiative funded by the Multiple Myeloma Research Foundation, which has sequenced over 200 tumors. His lab is currently focused on understanding how some of the most frequently mutated genes identified in his previous studies are involved in the pathogenesis of multiple myeloma. So thank you all for attending, and I'd like to welcome Dr. Jonathan Keats to begin his presentation.
Well, thank you, Andrea. It's a great opportunity to get to present to people some of the work that we've been doing within our group related to this exciting study, but also a lot of the background effort that's going on within our groups here at TGen to really make the study a success. As just a little bit of an outline, I'll give people a bit of background on what multiple myeloma is. A lot of people get it confused with melanoma. It is not a skin cancer. It's actually a leukemia lymphoma-related disease. And I'll give you a little bit of background on what the MMRF COMMpass study is and why are we actually doing that study. An overview of the assays used in the project, and then as part of and most related to this webinar, the internal testing we've done of various DNA library kits that have really led to our selection of currently the Kapa Hyper Prep kits that we're using across the board for all of our DNA-based assays. And then some of the early results from this study will give you a little bit of a teaser of what has actually came out of all that work. So for those that wouldn't know, multiple myeloma is actually a cancer of a terminally differentiated B cell, or which is properly called a plasma cell. So this is the B cell that usually homes to different organs and will secrete antibody. So it's what helps us fight off a lot of infections. So the easiest way I usually explain to people is if you look at what is multiple myeloma from a pathogenic view, if you look at a normal marrow, which is the picture on the left-hand side, you can see what a pathologist will call trilineage hematopoiesis, which for most of us would just mean I see a lot of different cells that look different. If you look on the other side, the myeloma bone marrow will be highly infiltrated with a single cell type. These are the large cells with large cytoplasm and an eccentric nuclei, which are the plasma cells. Or if you're my dad, you just call them sunny side up eggs. But one of the nice things of working on the disease and why we think is really a great disease to build personalized medicine approaches is it has an excellent biomarker. So because the tumor is the antibody-producing cell of the body, you can take serum from a patient and just do a very simple serum electrophoresis where we just separate the proteins based on their charge. And in a normal patient, you will see this normal band, which is really all the normal immunoglobulins we have that helps us fight infections on a day-to-day basis. In a myeloma patient, because a single B cell is now producing a massive amount of a single, or the tumor is producing a single antibody, all the different tumor cells, we get this large spike of the from the high protein load in the serum. As a consequence of the disease, which is kind of unique to myeloma, we do get this very severe form of osteoporosis, and in this X-ray you can see all these dark staining areas of the skull where the tumor is actually ate away at the bone. You can actually, it's almost like a baby's skull. It'll be nice and soft. So just to give you a little idea of how do people get diagnosed with this, well, in a good situation with the idea of early detection is best. Hopefully your yearly physical through a CBC, people would identify you have a high serum protein level, and that would lead to additional tests that may identify high calcium levels because of all the bone destruction, which releases calcium into your serum. Because the way we normally remove antibodies from our serum is through the kidneys. A lot of times when you have excess amounts, you actually start seeing protein in the urine. And then a lot of the patients, because the bone marrow is being replaced with tumor, you'll see anemia, potentially severe bone pain, and also recurrent infections. In a poor situation, the disease is discovered after a bone fracture, which is quite common. People have something snap and then go to the emergency ward and discover they have a broken bone and cancer. They'll have repeated infections, or renal failure will be another common presentation for some patients. Now, what I wanted to talk to you about is some of the very advanced genetic testing that we're doing, and it does help to understand what is the background of myeloma from a genetic perspective. If you step back to the 1980s when Cytogenetics was cutting-edge genetics, we were able to separate the tumors into two broad classes, one that we call hyperdiploid myeloma, the group on the right, which oddly has trisomies of many of the odd number chromosomes. And then the other group, which is another about 50%, which we call non-hyperdiploid myeloma, where we see more or less a diploid karyotype, though with some features like deletions of 13 and deletions of 14 being relatively common. Now, because it is a B-cell malignancy, one of the common mechanisms that we do see in multiple myeloma is rearrangements within the immunoglobulin locus, and I present this because I'll show a little bit of this data later. So a common mechanism is normally this VDJ segment is leading to the production. This is actually the coding sequence for the specificity of the antibody. And all these different C segments are what defines the IgM or IgD or IgG segments of an antibody. What frequently occurs in multiple myeloma is we'll have breaks within the switch regions as the cells are going through class switch recombination, leading to a rearrangement with another chromosome. And in this case, we're showing an example where normally this gene MMZ is expressed, but not at a very high level. But after the translocation, it gets brought in proximity to this enhancer. You get high expression of MMZ. And on the other derivative chromosome, this gene FGFR3, which is not normally expressed in any cells in the bone marrow, actually gets brought in proximity to this other strong enhancer and very highly expressed on the level comparable to actin. Now, as we went into the next level of technology with spectral karyotyping, what became apparent was not only were there these immunoglobulin translocations, but there was a lot of structural abnormalities throughout the tumor. And then when we went on to the level of doing SNP arrays and CGH with high-density array formats, what we could see is that a lot of those classic cytogenetic features were maintained and obvious, but a lot of very small deletions and amplifications exist in these tumors. But those two broad categories of hyperdiploid and non-hyperdiploid are still predominant. With the advent of gene expression arrays, many groups around in our field were able to leverage one of the advantages that we have in this field, which is not only is the tumor producing a biomarker, but the tumor is relatively easily accessed through a bone marrow biopsy. And the tumor has a specific marker on it which allows us to do magnetic sorting, so we can purify tumors to very high purities where almost everything we do for a genetic study would be greater than 90% tumor. So people have been able to very highly reproducibly generate classification schemes to identify different subtypes. So in this example, there's very clearly multiple different distinct subtypes of the disease. So pathogenetically, we would call it multiple myeloma, but we do split those out into what we would call the MS class versus the PR or proliferative class. So there are many different classes. The other thing that we've really gained from gene expression profiling was the ability to prognosticate patients, and in this example showing one of our risk models, the 70-gene risk model that's been developed, where unlike as we would expect, patients with high proliferation rate tend to have higher risk disease, but if you go through all the different groups, you can see some groups have higher amounts of high risk than other groups. And this does correlate with what we'd identify with patient outcome when we only looked at the subgroups before. Another thing that is work from my postdoc was starting to really look at how patients change with time. So we were able to, in this one example, follow a patient through their entire disease course, where we can follow the tumor content or tumor level based on the red and blue lines that just mark the burden of tumor at any given time point. And then the different therapies, which are the color bars that the patient received. And in this patient, at multiple time points, we did FISH, CGH, whole genome sequencing, among other tests. And what came out of that is was that we were able to show that this patient actually had multiple clones that ebbed and flowed in what we have termed the clonal tides as the tumor was attacked with different therapies. And the last thing that I really need to present, that it kind of brings things, because as you've seen, there's a lot of genomic data in multiple myeloma already, but what has happened over the last decade has been nothing short of amazing. That the survival of patients has more or less doubled, verging on tripled in the last decade, going from what were cutting-edge studies in the early 2000s where median survival was about 4 years, to the current time point where median survival for most of our studies have not even met outcome at 8+ years. So the survival has been dramatically increased, and this is largely to what in the field we term the novel agents, which are really two classes of chemotherapeutic agents. One that is the proteasome inhibitor, so the first-in-class molecule is bortezomib from Millennium, and then more recently, the most recently FDA-approved one, carfilzomib from Amgen Onyx. And these, we think, really target the unfolded protein response in these cells that have high protein load, and they're just teetering on the verge of being able to tolerate that high protein load. Then the other class is what has been termed immunomodulatory drugs, which are actually based on the drug thalidomide that caused all the birth defects in the 1960s. This drug class has recently been shown to be functional through its binding of a protein called cereblon, or CRBN, which changes the E3 ubiquitin ligase specificity of this complex to now actually degrade a gene that's essential for plasma cell survival, Ikaros and Alias, or IKZF1 and IKZF3. So with that in mind, that led to the development of the MMRF COMMpass study. And it really was with the idea that we have a lot of good genetic data, we have a lot of classification schemes, we have a lot of prognostication, but all those things were based on historic treatment regimens that are rarely used today. So to really provide the field with a solid basis to build a personalized medicine initiative, we wanted to be able to investigate a large cohort of patients. And this has really been a massive task that has brought on, what you see in the blue dots are these sites all over the world that are part of this study. So what is the study, so people have a better idea? The goal of this study is to accrue 1,000 newly diagnosed patients with multiple myeloma. Then those 1,000 patients are put onto 3 different arms of the clinical trial. One that requires they go on to an IMiD-based regimen, another arm which is proteasome-based, and then a third arm where patients can go on to IMiD and proteasome-based, so the combination of the two. And then to really understand how the different therapies involved modulate how the tumor evolves with time, we're also going to actually characterize 800 relapse events from that cohort of 1,000 patients. Importantly, it is actually a clinical trial, so we do accrue a full clinical workup of every patient every 3 months, and every patient is followed for a minimum of 5 years. There are a number of clinical assays that are being done and provided to the clinical sites, largely because flow cytometry to characterize the tumor, but also as minimal residual disease is very prominent in our field. We do some DNA content that does help identify that hyperdiploid subtype. And up until recently, we were actually doing BRAF V600E mutation detection in a clinical lab, and that's just now moving to a more high content assay. So what I wanted to kind of go over is what we're actually using as our characterization model for this cohort of patients. So we do 3 different assays here at TGen for every patient sample and tumor sample that comes through. We do what we call a long insert shallow genome. Now this is a 1 KB insert through a standard Illumina library that we sequence with the goal of being able to identify copy number changes and also structural changes. The important thing here, particularly for structural changes, is because of the long insert, we get about a 8 to 7x base coverage genome, but the physical coverage that is powered to detect structural rearrangements is equivalent on average for most of our patients close to a 60x whole genome. For exome capture, we'll go into a little bit deeper what we're using, but it is a 75 megabase capture that we use, and currently our median coverage is about 110x. And then for RNA sequencing, we're using mRNA sequencing, and I won't go too detailed into much related to the mRNA sequencing, but due to some of the issues with the high amount of immunoglobulin RNA that we deal with, we do attempt to go very deep on these patients, and we're pushing upwards of 200 million reads in each patient. And really, the central dogma between everything that we're doing is following the central dogma that if you have an abnormality in the DNA, it should be evident in the RNA and should hopefully produce a mutant protein that is having a biological effect. And by integrating all these different data streams, that we should be able to very comprehensively understand what is going on in each tumor. So one of the things when we started this project, we started actually using TruSeq exome and TruSeq DNA kits from Illumina. After about a year of the study, Illumina chose to discontinue those products. So we started actually testing a large number of products to see what we would switch to replace that discontinued item. And we went through a number of different tests. We tested different DNA library preparation kits. We tested library amplification enzymes, and I won't go into much detail on those, just to say we had elected to use the Kappa Hi-Fi enzyme actually before we had even started testing the library kits. And then for exome capture, we settled on using the SureSelect V5 plus UTR for this study. And again, I won't go too much detail on all the testing related to those, but for library preparation kits, we'll go through some of ours. So just to give you a little idea, we're actually pushing upwards of batch 31 now for this study. We get batches of samples from our biobank every month. So for batch 1 through 10, these were all done with TruSeq kits with 1,000 nanogram inputs for the long insert and for the exome. As we moved into batch 11 and 24, one of the issues that we had identified already in those early days was that a large number of samples were not getting analyzed because we didn't have sufficient material, so that we had a large push to try and move to lower input protocols. So in testing, and we'll show you some of the data, but we eventually selected to use the Kappa with bead protocol, where we were able to decrease our standard inputs down to 500 nanograms, and then 250 nanograms or 150 nanograms for the exome long insert respectively when we had less material available. And then more recently, and we'll again show you some of this data, we've moved the long inserts and exomes to 200 nanogram input as our standard input using Kappa Hyper, and 50 nanograms for the exomes when material is limiting. So I'll just walk you through some of our initial testing as we move to the Kappa library kits, and really again to just reinforce, the goal was to identify a library kit that could support whole genome preparation for both exome sequencing and long insert sequencing, which has some somewhat, mostly similar, but some distinct issues that we wanted to resolve. In the testing, again, we started with our classic input of 1,000 nanograms, using then also testing down to 500 and 200 nanogram inputs. And these all went into, for the exome, standard Agilent XT2 preparations, which at the time we elected to use. To keep everything consistent, we used adapters from Agilent, which were their full-length XT2 style adapters. We also tested the NAB Ultra kit, and again, we used the Agilent XT adapters to keep everything constant. We used pre-capture pooling, and these were sequenced 100 by 100 using Illumina technology. But it doesn't take long to recognize that if you look at this slide, if you look at the two columns related to the Kappa on bead compared to the Agilent XT2 prep and the NAB Ultra prep, that when we provided 1,000 nanograms or 500 nanograms, the total quantity of library that was produced, and again, this was standard number of PCR cycles, standardized inputs, that the Kappa with bead protocol almost doubled the amount of DNA or library produced compared to the other kits. And from our view, it was simply that if we're putting in the same amount of DNA and producing more a library, that this kit must be more efficient for some reason that we couldn't attach. Just to show you the next level, after just how much DNA do you produce, but after you actually sequence the library, and you look at the number of unique library molecules that were produced, and this is actually calculated from Picard marked duplicates, you can see that even with the Kappa with bead protocol at 200 nanograms, that we were actually producing more unique molecules than the Agilent kit, which is shown here at 1,000 or 500 nanogram inputs. So based on these studies was what led us to select that kit for exomes, but we wanted to confirm that this was working also for our long insert protocol. So we tested a series of cell lines, and what I'll walk you through here was that we took 2 cell lines, one JJN3, one KMS11. The 2 of them were compared with the Kappa with bead protocol to our classic TrueSeq protocol that we'd used, more with the idea of saying, do we at least get comparable data? And then for KMS11, we actually compared the Kappa with bead protocol and also the Kappa protocol where you don't do all the steps with the beads in the slurry. And what we were able to show is that consistently the Kappa protocol led to a lower duplicate rate. Now these are relatively shallow whole genomes, so any significant change there is of some significance to us. But also, and this is largely more related to the Kappa Hi-Fi enzyme, the GC dropout rate was dramatically lower in those libraries. So we had pretty good confidence that we could now migrate to Kappa with bead for both of the 2 assays that we were interested in. Now to give you a little idea what those libraries look like, so comparing and we have a 5-letter code for everything, so if you look here on the left-hand side, the KBWGL is Kappa with bead whole genome long insert compared to TrueSeq whole genome long insert. We get very similar profiles after the gel punch. But if you actually look at the GC content distribution of these libraries, it becomes very quickly apparent that this normalized coverage plot for the Kappa library, pretty much you have nice normalized coverage across the entire spectrum of GC, while this plot, as soon as the GC content exceeds more or less 50%, the coverage dramatically dropped off. And what was a little bit frightening to see with that was when we went into this test set where one of these tumors has a known IGH translocation, what you can see is a lot of these translocations do happen to occur in high GC-rich regions. And you could see 2 things. This region where the actual breakpoint that I'd previously cloned as a graduate student, you could see that there were a couple of reads that were informative of the break, but very little evidence. By comparison, the other 2 libraries done with the Kappa on bead or the Kappa standard kit, there was ample coverage to define the breakpoint. Another interesting thing, and just so you can see over here, another start code or the initiating exon for this gene WHSC1, huge island of no coverage because of the excess GC coverage, versus with the Kappa library kits we were able to get coverage in those regions. So not only were we producing more library, but more comprehensive and more uniform libraries. So what were some of our initial conclusions from those studies? So the Kappa with bead library kit outperformed the library kits that we had tested, produced more complex libraries, produced more uniform libraries. And this change decreased the amount of DNA requested from our biobank from 3,000 nanograms down to 750 to 1,500 nanograms, which had what from my side is the most important effect. It increased the analyzed number of samples from about 34% to 58% for this cohort. And again, if you consider we're trying to accrue 1,000 patients onto this clinical trial and then analyze them, well, if we get 1,000 patients on trial and only analyze 340, versus if we get 1,000 patients on trial and can analyze 800 of those, that's a much more efficient usage of the study dollars that we were trying to use. So even at 58%, we weren't happy, so we were fortunate that Kappa was able to allow us to get early access to the HyperPrep protocol. And here I've just kind of provided a little bit of an overview of what the protocol is. Compared to a lot of the other standard library kits that have been around for the last couple of years, the end repair and air tailing is actually in 1 step. And interestingly, one of the major changes is that the A-tailing reaction is done at a higher temperature. And we'll show you a little bit of data where we think that has a huge effect. And then the cleanup steps, there's been a couple of cleanup steps that are removed that really speed up this whole process. One thing I will comment on is due to the volume that we do within my group, we have migrated most of these protocols, both the Kappa with bead and the Kappa Hyper, onto the Agilent Bravo automation system. So most of the data I'll show you has been done on the automation platform. What I would say is we've done a lot of tests, and you'll see coming a little bit, to try and even optimize those automation platforms to get data that's equivalent to hand preps on the automation systems. So what I want to talk you through was some of the first rounds of testing that we did with the Kappa Hyper kit. Again, to make this really consistent, these were all done on the Bravo, so well-to-well reproducibility should be very high. 2 columns were set up with inputs of 200 nanograms, with everything being in duplicate. And we tested different adapter concentrations, and again, this is using Agilent XT2, or what is a full-length adapter style. So we used the classic 1 to 10 concentration of adapters, 1 to 20, 1 to 40, and 1 to 50. Then all these were amplified together. We did the 2 different columns at 2 different cycles, so we could actually calculate the amplification efficiency, and I'll talk to you a little bit in a second about why that's important. And then all amplifications were done with the Kappa Hi-Fi and actually the primers that are provided with that now. So what you'll see here is what we were able to show is that increasing the concentration of the adapter from 1 to 10, which is our standard methodology, up to 1 to 20, 1 to 40, 1 to 50, led to a linear increase. So obviously the most dramatic effect is seen at the 1 to 10 to 1 to 20 when you make that initial doubling. Because we did this cycling at 2 different concentrations, or 2 different time points, a number of cycles, and assuming that the efficiency of amplification is consistent cycle to cycle, we were able to calculate that the PCR amplification efficiency was 65% in this, and I'll show them a little bit later. We've repeatedly seen numbers when we do these tests between 65 and 72%, so that can be a pretty standard assumption for most library amplifications, I would assume. But what we were able to do is, looking at this, actually look at, because we converted the number of cycles, so we could actually look at the percentage of input DNA that's converted to library, and this is actually one of the calculations I think is extremely valuable for people to start using, where you calculate the amount of input DNA that you had related to the amount of library molecules created, so you can get an appreciation for what percentage of the input DNA was converted to library. And what you'll discover when you do those things, as we discovered, is library preps that we had previously done that generated very good sequencing data will actually have conversion rates of in the range of 1% quite often. And what you'll see here is that even at 1 to 10 with the HyperPrep kit, we were up at conversion rates above 10%. And when we got up to 1 to 50, we were in the mid-30s, so about 35% of all the DNA that we put into the reaction actually got converted to library. And if you extrapolate that out, it theoretically will keep going up and up and up. And this has led us to doing things where we're now making our own adapters so we can have higher concentration adapters, because as we increase the concentration of the adapters, we've consistently seen this increase in the conversion rate with no increase in adapter dimer formation. So then the second round of testing we did was to bring it more in line with actually our exome preparation. So the long inserts are prepared with full-length adapters, while the exomes now are produced with short adapters to go into the Agilent XT workflow. So similarly, we set it up exact same experiment, 200 nanograms each, 2 columns each, 1 to 10, 1 to 20, 1 to 40, and thanks to the difference in the concentration of the adapters, we were able to go up to 1 to 80 relative to the recommendations. These were amplified with 8 cycles and 10 cycles, so you notice an immediate thing that the amplification, or the number of cycles necessary with the short adapter versus the full-length adapter, is noticeably higher, and that's been something we've seen consistently regardless of kit used. And one of the things that we'll caveat is that we increased the amount of amplification primers provided by Agilent to increase the efficiency. What you'll see again is a very similar plot here, that as we increase the concentration of the adapter, that the amount of library produced increases consistently, and again we were able to show that 65% amplification efficiency. And what you'll notice here, though, is that when we convert that to the percentage of input converted to library, with this shorter adapter, it was noticeably less, so that even as we got up to 1 to 80 adapter concentration, we were only pushing up around 7% of the input DNA was converted to library. Now, what I'll point out to you is if you consider a lot of our previous preps that had been done were in the 1% range, this was still a major increase. But it left us having this feeling that there were ways we could still improve this dramatically if we played with it a bit more. And just to give you a little bit of idea, if you compare using a full-length adapter, which is XT2 style, or the red bars, versus a short adapter, the XT style, what you can see is with the full-length adapter, the conversion efficiency with the same amount of input, done on the same robotics platform with the same protocol, does lead to dramatically higher conversion rates. So based on that, we went back to the drawing board a little bit and reviewed some of our protocols. What we discovered in doing that was there were some tweaks we could make to the automation protocol that increased the efficiency with the Kappa on bead significantly. 2 things on the Bravo, plus also making some changes to how the amount of amplification primer we provided. And then because we'd seen this linear increase in the amount of adapter, when we decided we would just jump right in and do a 1 to 300 input to adapter ratio. So this is magnitudes higher than most people would ever do with a previous kit, where you'd be worried of a huge excess of adapter dimer formation. What we did there is now you can very clearly see is this, with the hyper prep using XT style adapters, we were able to actually push the conversion rate up to in the area of 20%, which beat any of our previous work, even our optimized Kappa on bead protocol. So based on these results, we decided that we would actually move to using the hyper prep kit across the board. Now, what else is important is what we've kind of shown there is that at 200 nanogram inputs, we are basically able to generate libraries that are significantly better than anything we'd generated before, regardless of input. So that's at 200 nanograms as our base for what we'll do all library preps going forward. But to continue to increase the number of analyzed patients, we wanted to see how low could we go. So this is just, again, another set of experiments showing that when we did some exomes where we used 10 nanogram inputs or 50 nanogram inputs into Kappa Hyper, again, this is with flooding the adapters at a 1 to 300 ratio, that we were able to, with the 50 nanogram libraries, maintain that roughly 30% of input converted to library, so nice and linear from 200 nanograms down to 50 nanograms. The 10 nanograms, we weren't able to show the same conversion rates. We were in the 5 to 10% range. So that still seemed acceptable to us, so we took those on to sequencing. And just looking at these, we actually sequenced these quite deep, leading to the mean target coverage for the 50 nanograms were in excess of 150x. But even the 10 nanograms, after removing the duplicates, the coverage of these exomes was 75 to over 100x coverage. So though there was the percentage duplicates definitely jumped up into the 50% range for those 10 nanograms, we were able to generate by what would by most standards be considered a very high quality exome, even with 10 nanograms input. But more importantly, with 50 nanogram inputs, we were able to generate exomes that, though their duplicate rate is a little bit significantly higher than we see at 10 nanogram or 200 nanogram inputs, still generated data that was perfectly usable for our purposes in this study. So that led us to the next set of conclusions, which was that the efficiency of library production is significantly improved compared to other products tested when you use the Kappa Hyper Prep kit. Efficiency is highest when full-length adapters are used. We're still trying to really get a good understanding of that. The adapter quality does seem to be a huge contributing factor. So we've started playing with building our own adapters, synthesizing them and making our own adapters. And we have actually seen dramatic increases. So with full-length adapters, it's quite possible to be pushing upwards of 60% of library molecules will be converted, or input DNA will be converted to library. And these results were really the basis for our justification to switch to the Kappa Hyper for our exome and long insert preparations using 200 nanograms as our base. But importantly, even for the exomes, we're now going down to 50 nanograms. And the important part there is, as I mentioned, again, that 1,000 patients are being accrued onto the clinical trial, and we want to analyze in a perfect world all 1,000 patients. Over the last year, since we've implemented these changes, we've now pushed our percentage of analyzed patients up to 89% for the cohort. So these changes have dramatically increased the number of analyzed patients in this study. So that kind of overviews what we've been doing when testing the Kapa Hyper XT. So I thought I'd give you a little chance to see some of the data that we recently presented at the American Society of Hematology meeting a couple of weeks ago related to this cohort. So for those people that are aware of studies like the TCGA studies and ICG studies, you'd be aware that those are very large cohort studies. Most people may not have previously heard of this COMMpass study, but as you can see, the goal is to get 1,000 patients, and we expect to have 1,000 patients fully accrued and sequenced, or those sequenceable by the middle of this year. But what we can talk about today is this cohort that we released in January of 2014, which had 436 patients that were on study, 192 untreated sequenced patients with 5 relapsed patients being sequenced, and a total number of patients sequenced at 196. What is a unique model, because this is not a government-funded study. It's, again, funded by the Multiple Myeloma Research Foundation. So they rely on philanthropy from donors, but they've also been able to sign a pre-competitive consortium deal with a series of pharmaceutical companies that we term the PMIC. And these people get access to the data for 6 months after it's generated here at TGen. Then for the following 6 months, it's available to the 89 clinical sites that have contributed samples. And then after that 1-year window, the data goes fully public. And there's 2 ways you can get public access to the data, and you'll see this again later in the last slide. But they're at research.mmrf.org. There's a beautiful portal for accessing the data. But also the raw BAM files have all been deposited in dbGaP. And as you can see, we'll be incrementing this forward in the very near future, where, again, over 300 patients will start becoming available for most people to see in the near future. So one of the things we focused on, again, what makes this study a bit unique is that long insert technology. And as we mentioned, there are frequently translocations in the immunoglobulin region. So we've zoomed in on the immunoglobulin heavy chain region here on 14q32 on the very terminus of chromosome 14. And we've been looking to see which translocations we were able to identify within this region. What you can see is in the colored regions is all the translocations that are known to exist. So rearrangements targeting FGFR3 and MMSET, as I previously showed, rearrangements targeting the MYC locus, rearrangements targeting cyclin D1, rearrangements targeting the MAF oncogene. What was interesting when we looked at this study was 2 things. One, historically in our field, these green rearrangements that target MYC have been associated with as progression events. They're relatively undocumented in diagnostic patients, which this cohort is exclusively at this point largely. And what we were able to show is that these rearrangements do exist at diagnosis, and it was largely because the previous assays that we had done had focused on rearrangements that were centromeric of MYC, not telomeric. And what we've been able to show with the RNA sequencing data is all of these patients with these rearrangements do overexpress MYC. So these are definitely translocations targeting MYC. So our clinical assays will need to be resolved. The other thing you see is what we've tried to mark with some of these stars are that there are a number of translocations that seem to be going to somewhat random regions. And what we've been able to show is the vast majority of those are actually markers of complex events with other canonical translocations, and I'll show you an example of those in a second. But there are also some patients who have recurrent rearrangements with this gene MAP3K14, which is a gene that helps regulate the non-canonical NF-kappa B pathway. Previous work from myself and a couple of other groups have previously shown that dysregulation of the NF-kappa B pathway is present in about 30 to 40% of patients through a variety of mechanisms. But this is one of the first recurrent translocations that we've been able to identify in this cohort. And then there are also a number of translocations that seem to coexist when a canonical one also exists, suggesting that sometimes there's not just one translocation in a patient, but there can be multiple in a patient targeting the immunoglobulin locus. So as an example of one of these complex rearrangements, we can just walk through what this is, again, each long insert read being shown with a piece of software called Periscope that's available from WashU. You can follow down the centromere of chromosome 14. So the strong 3' enhancer would be down here. If you follow this down, you actually jump from this part of chromosome 14 down to this part of chromosome 11, which over in the distance is cyclin D1, which is highly overexpressed due to this translocation. And then if you follow the other centromere starting in chromosome 11, it actually takes a little detour, jumps down to chromosome 19, picks up a couple of kilobases of chromosome 19, and then jumps back up onto chromosome 14 over here, leaving this area in the middle actually absent or deleted as a consequence of the rearrangement. And again, a lot of these things, what we've seen with these is this is largely detectable with the long insert technology. And what you'll see here is this region here where you see less evidence is largely because this region here, where proximal to the breakpoint, is leads to mapping errors because of how repetitive it is in the genome. So not only is long insert powered to detect rearrangements, but it's also very powered to deal with complexities of the genome compared to shorter insert methodologies. The other one I wanted to show was one of these cases with a simultaneous dual IGH translocation. So it does look like in this case that two independent translocations occurred simultaneously. So we have a classic, again, T1114, where, again, you start over here on the centromere, walking down, and, start here on chromosome 14, walk down the centromere, you jump back up, and now we target the dysregulation of cyclin D1. But the other derivative chromosome was also created, so starting on the centromere of chromosome 11, walking down the chromosome, taking a ride down to chromosome 14, producing the classic derivative 11 chromosome. But at the exact same point where these breakpoints were occurring, there's another breakpoint that actually is jumping over to chromosome 20, just downstream of BCL2 like 1, and it's leading to the overexpression of this gene. And importantly, what was nice by having matched RNA-seq in this case, we were able to show two things, that the gene is overexpressed. So here you can see that BCL2 is highly expressed in this patient, as is cyclin D1. But what also came from those studies is not only was that gene overexpressed, but we were able to show that the anti-apoptotic isoform was actually the prominent isoform that was produced through splicing. So the next thing that we had focused on in this study was to, again, look to see if there were any immunoglobulin fusions that were detectable in these patients using the RNA sequencing data that we haven't talked too much about. And what we were able to show there is that there were two recurrent fusions detected, one that's been well characterized in the field, target where immunoglobulin heavy chain elements coming from chromosome 14 are actually associated with WHSC1. This has been well documented in the field. We do see some rare rearrangements of, again, the immunoglobulin heavy chain region going to MYOV. We believe this is just targets of the cyclin D1 translocation, as the same patients all had breakpoints defined by long insert technology at the same points. But what we were able to do was leverage the fact that we had the coexisting long insert data to filter out a lot of the noise that comes from the fusion detection software and only look at fusions where there was evidence for the fusion in the RNA but also in the DNA. And then we were able to look to see, are there any partners within the genome that have multiple targets? And what came out of that was, first off, the most common one we were seeing was fusions involving MAP3K14, that same gene we'd showed had IGH translocations previously. And then another gene, this RGEF and FCHD2. These are both highly expressed genes. We think they really represent promoter replacement events where highly expressed genes within these myeloma or plasma cells are brought in proximity with potential putative oncogenes and drive their expression to high levels, leading to the dysregulation of the pathway of interest. The other thing that we noticed from these studies was that a lot of these deletions, these ones shown in purple, this showing CDC42 binding protein B, these were actually coinciding with deletions of this locus that we do. Again, we previously shown targeted a gene called TRAF3, which is part of the NF-kappa B pathway. And we've seen this repeatedly now, that a lot of the quintessential tumor suppressors are actually being targeted through fusion events. And we believe this is just another mechanism of inactivating a tumor suppressor where you, instead of deleting it or mutating it, you break it in half through a translocation or a structural event that then inactivates that allele, providing another inactivation event. And sometimes when you have those structural rearrangements, you see fusions, and sometimes when you have the deletion that targeted a gene, that gene or bystander genes will show fusions. So we found that the fusions can be very powerful for identifying common regions harboring a tumor suppressor gene. Now, what else has been very valuable in this study compared to a lot of the other studies is that, again, almost every single patient has matched RNA sequencing and exome sequencing. So just as an example, what you quickly learn from these kinds of studies is that most of the mutations that we talk about are discovered at the DNA level. They are never expressed. And again, this largely has probably two sources. One, if you look at an individual cell type, only on average, probably 50% of the genes in the genome are expressed in any given cell type. Not every gene in the genome is expressed in every cell type. So that's part of the reason why we see this. But what we'll clearly see is that most of the mutations, the allele frequency in the DNA tends to mirror what we see in the RNA, which was nice to see. We do see some evidences of cases where, in this case, where a relatively highly expressed gene, we do see that the allele frequency in RNA is less than we do see in the DNA. And in cases like these ones up in this quadrant, where we do see that the allele frequency in the RNA is much higher than we see in the DNA, where there's a bias toward the mutant alleles expression. What that's allowed us to do is be fairly aggressive in the mutations that we've identified through the exome sequencing procedure and filter out a lot of the things that, though tools like, say, MuteSig from the Broad Institute may flag the gene as being significant based on the pattern of mutations, we were not able to show that that gene is expressed in any patients and the mutant allele is not expressed in any of the patients. So we've removed genes like that from the list. And this has allowed us to provide a very high confident mutation list from these studies. What's been very nice to see in the myeloma field is that, though similar to most of the cancer sequencing studies where genes like KRAS and NRAS are very prominent in their frequency, we do have a couple of novel genes, so FAM46C and DIS3, which are two genes we previously identified in the Myeloma Genomics Initiative, are highly currently mutated in this cohort and other myeloma cohorts. TRAF3 gene that we had previously identified as being, at that time, the most commonly inactivated tumor suppressor in the disease, and it still remains close to being that. And then BRAF, which had been identified in that most recent study. But we do have a number of additional genes that are slowly slipping up on our list of important genes to focus on for the field in the future as we try and develop personalized therapies for these patients. Now, again, one of the important things for this study is the goal of this study is, one, to characterize the patients at diagnosis, but also to really have a good understanding of how these patients evolve with time. So in this case, we have a diagnostic sample, then followed by chemotherapy treatment for 6 cycles, and then maintenance treatment. And then the patient eventually progressed 21 months later. We were able to sequence this patient and show a couple of interesting things. There are a number of genes that were mutation frequency is relatively constant at both time points, suggesting some of these are likely the initiating event within this patient. There are a number of events that are lost at relapse, and what I'll focus you on is that most of these are these dark blue plots suggesting that the gene is not highly expressed. So most of the gene mutations that are lost are in genes that are not expressed, which would make some sense. They're less likely to be required. Interestingly, most of the genes that were accrued or appeared at relapse are actually expressed, suggesting a reason for their enrichment. Interesting from a clinical perspective, given the availability of BRAF inhibitors, this patient was actually BRAF positive. At diagnosis, the BRAF clone was a minor percentage. It was only 36% of the cell fractions, while at relapse it is increased up to almost 90%, suggesting it may be a viable target at that point for this patient. Now, what I wanted to provide to people was just, if you are interested in this study, there is an excellent way to access all the data, the clinical data, the flow cytometry data, the sequencing data, the survival data. And this is at the research.mmrf.org. It's available to any academic person free. We also do provide the data, so the raw BAM files for all the sequencing effort is actually in dbGaP under PHS 00748 that people are free to access. And again, every 6 months we increase, pretty much double the data in that accession. What I did want to just take a quick second is to acknowledge many of the people involved in this study. as related to the goal of this webinar, which was really to present a lot of the effort that we've done characterizing library kits, that has been done principally in my group, largely through a very excellent technician in my lab, Christy Stephenson. She's been really driven 90% of that effort. And then Winnie Liang, who heads our sequencing center here at TGen, and her team, Lori and Jackie and Jonathan and Adrian, they have also dramatically contributed to the evolution of these protocols as we took them out of a testing phase and put them into production. Without that team of people, none of this work would have been possible. And I think we'll take a minute break and let Andrea take back over so we could answer any questions that people have posed. Thank you very much.
Excellent. Thank you so much, Jonathan. And actually, we have about 6 or 7 questions. Thank you all for submitting them. And we are ready to actually go. We've had them all set and queued. So, Jonathan, the first question that came through was, what are the types of samples and the number of replicates for the Kappa versus TruSeq comparison? Was it just exome sequencing, or did you also perform whole genome sequencing prior to exome?
For those, so the input material is coming from fresh frozen material. All of our DNA isolations have been done with QIAGEN pure gene preps, which is a thing we didn't talk about that dramatically increases the amount of DNA recovered in our hands anyways. For the replicates, most of those were 2 to 3 sample replicates, where the samples in those testings are relatively limited. For the libraries that were used to go into exome capture, we only sequenced the exomes. We didn't sequence their corresponding whole genome libraries. But again, all those tests were simultaneously done for the long insert, which is a whole genome screen, and those were also sequenced. So generally, it's been 2 to 3 replicates, mostly triplicate replicates for those studies.
Excellent, thank you. So the second question is, what was the % of patients analyzed prior to your optimized protocol? And do you think the increase to 89% is attributed to the library prep, or were there other variables that you didn't mention?
Yep. So our optimization has been twofold. I guess the very first step was optimizing DNA RNA recoveries, which were from the very first couple of months of the study, where at that point we were using QIAGEN all-prep kits, which produce relatively good RNA, but their % recovery for DNA is very poor. So we eventually switched from using all-prep to actually doing independent RNA and DNA preps, where the DNA is prepared with QIAGEN pure gene preparations. That we didn't mention, but that had increased our % analyzed from about somewhere in the 25% range to the 35% range that we mentioned. All the increases from about 35% of analyzed patients and up has been through improvements on our side with the molecular inputs, and that to our side has all been related to being able to decrease the amount of input material. We do have a lot of patients where we do only recover 1 to 2 micrograms of material, and then particularly as we get into these relapse cases where the sample availability can be in the hundreds of nanograms and sometimes lower, unfortunately. So the increase from 35% up to the 89% has largely been due to improvements in library methodologies that allow lower input. And what I would focus on is though lower input has been around for a while, we really stuck to our guns that if you did a lower input, it had to generate data quality comparable to the higher input. And in our hands and our belief, particularly our 200 nanogram with hyper preps, those have been generating quality that exceeded anything we previously done, quite honestly, even with the Kappa with B protocol. Okay, and somewhat related, so a question that actually just came in, and I'll ask it ahead of another, is what is the percentage of plasma cells required for exome sequencing? So as part of this study, again, we purify using anti-CD138 microbeads on a magnetic sorter the tumor from the whole bone marrow aspirate. In this study, any sample that has been submitted for sequencing showed an excess of 80% plasma cells. We do, for some of the samples, We will do the exome if it is lower, where we actually increase the coverage for those, but those are rare cases. So the vast majority of these samples, like 98%, are 80% plasma cells of what was after sorting. And most of them average over 95%.
Excellent. Okay. So what percent of the long insert reads are chimeras? What is the maximum percent chimeras that allow accurate determination of translocation?
Yeah, so I can say two things. One, for this study, we don't release data where the chimera rate is above 3%. So that is our cutoff. Since moving to the HyperPrep kit, we have not seen a single one go over 1%. And that's been one of the hidden magic sauce, and no one will tell me what it is for the HyperPrep kit, that we can increase the amount of adapters and not get adapter dimer formation for some reason. But more importantly, the chimera rate has literally been flatlined at under 0.5% in our exomes and our long inserts ever since switching. So there is something unique to the HyperPrep kit that literally blocks chimera formation. My personal assumption is that the A-tailing reaction with whatever change has been made is so much more efficient. It just literally, by having more efficient A-tailing reaction, you're not seeing chimeras. But I hope that answers the question.
Excellent, thank you. So if conversion rates are low for libraries, can you overcome this with additional sequencing, or are things just missed because of low complexity?
Well, obviously, if you want to do enough sequencing so that you completely saturate your library, so if you really had only 100 million molecules and you sequence to saturation, you're going to potentially cover it. Again, what I would point out is a lot of the work that we've done previously, we were seeing, like literally it was disturbing to see it was in the 1% to 2% range what our conversion rates had been historically. And those generated what we thought were very high quality exomes with duplicate rates in the 10% range on average, I would probably say. So I don't think it's a hindrance, but obviously the advantage of having a more complex library is you don't have to do as much sequencing to get to the same point, and that's why we've been focusing on that, is to try and either do one of two things: do the same amount of sequencing and get higher levels of coverage, which is what we've elected to do in this study. But in other studies, by increasing the complexity, we've been able to get to the same point with less sequencing.
Excellent. And our last question, right on time. So have you done any work on circulating tumor cells, if any, in multiple myeloma?
We here at TGen currently have not, though we do have a principal investigator here who is principally focused on doing those kinds of things, and I know we've been bringing him into our discussions on this, but currently, as part of this study presented here, the COMMpass project, or what's going on in my lab, no, we are not doing that today.
I do have one last one, and I know that was kind of a quick question. We'll try it quickly. So what is the percentage of failure getting 80% or more plasma cells from relapsed patients?
From relapsed patients who are in a bona fide relapse, it's really been similar to what we see in our newly diagnosed cohort, and that would put it in about the 5% range. That samples that we had a good quality marrow specimen. Generally, it's pretty rare that our samples have less than 80% plasma cells. Again, I don't have a distribution plot there, but we do see that, again, the median for our plasma cell purity is, I think, 96% from our different tests. So dropping below 80% is relatively rare. Actually, being in the 80s is relatively rare, quite honestly.
Excellent. So that concludes our webinar, and we missed the last two questions, but fantastic. Thank you all so much for joining us. Thank you, Dr. Keats. We're really honored to have had you with us for this and collaborating on this. If anybody has any questions or would like more information, please feel free to reach out to us at kapabiosystems.com, and my email address on the original registration confirmation. Thank you again, and we look forward to presenting to you in the future.
All right.